This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 411, recorded on October 14th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hi, Vincent. How are you?
Very well, thanks. Gorgeous day here. Man, blue
that's
skies.
Good. What do you got?
We got blue skies, not a cloud in the sky. 16 Celsius, 41% humidity, and it's very crisp. You know, you can see far because there's not a lot of humidity in the air. That nice. Fall air, beautiful. Leaves are turning here.
We have partly cloudy, 84 Fahrenheit, 29 Celsius. It's a nice day. The mornings are getting cooler here, so we're getting into fall.
Now, in Austin, it's going to be a little cooler in the winter, right?
Just going to look that up. Yes, I was going to see what it is in Austin right now. Yes, it's going to be cooler during the winter. And my guess is, hold it, hang on. Wait for it. Oh, yeah. Actually, right now it's 82 in Austin. So it's about like it is now.
Yeah. Well, it's farther north, so it should be a little cooler.
Yeah.
Right?
So...
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 61 degrees here, which is 16 Celsius. You remember 61 is 16.
Yes.
Same as here.
Not a cloud in the sky. Just gorgeous day.
Nice. Also joining us from western Massachusetts, Alan Dove.
Good to be here. And it is virtually identical weather here. It's 61/16 and clear blue sky, just a lovely early autumn New England day.
Doesn't Alan have a great sound?
Oh, thank you.
That's the microphone plus his
the new
voice.
mic. It's the mic you sent me.
But definitely, you got Alan, Rich, and Kathy. You should consider having me send you a new microphone, and you can sound just like Alan. Well, not quite, you know.
You'll still be able to sound.
Yeah, still like me. But yeah. Sure.
I will seriously consider
microphone.
Yeah. Wait till I'm settled.
I'll wait till you go. I'll wait till you go because it's. I know, Kathy, you've had problems with your ears hurting after a while with the headset.
With the top of my head.
Yeah, that's
and my ear a little bit. Yeah.
Yeah, with this way you won't. We'll give you a nicer headphone, and you won't have any pain, and your voice will have. What is the word for in music where fullness? Is there a Latin
resonance?
Word? Resonance, etc.
Maybe. Yeah.
Not that it's bad now; it's actually quite good. But it might be, it'll be better and it'll feel more comfortable.
Yep.
Cool.
We have a winner of the third giveaway of the book Viruses by Marilyn Rusink. Remember, 2 TWiVs ago, 409, we said the 50th emailer will win. And the 50th email arrived at 8:20 a.m. on October 11th, which I believe is Monday, this past Monday.
Tuesday.
So 9 days after the episode was released, in case any of you would like to calculate. And I just got some in today. We got over 67 submissions total. The winner is Rich Siggers, who initially wrote, simply said, TWiV is amazing. So I asked him for a little more, and he wrote, thank you very much for the email. My colleagues, who are also TWiV listeners, were both very excited and a little jealous that I was the lucky winner. They were particularly jealous when I said I was going to ask you to sign the book. A little about myself. Firstly, and most importantly, I am the father of 2 amazing boys, Jack, 10, and Lucas, 8. Other less important things about me include the following. I did my undergrad and master's at University of Saskatchewan up here in beautiful Canada, where temperature is only measured in Celsius. I then completed my PhD and postdoc at the University of Copenhagen, Denmark, before moving to Pittsburgh, where I was an assistant professor of research at the university in the pediatric surgery department. While I love my time in Pittsburgh, I could never make the flip from Celsius to Fahrenheit. After Pittsburgh, I moved back to Canada, began working at Health Canada, similar to the FDA in the U.S. I work in the area of vaccines and consider TWiV an excellent source of information and a great addition to my literature reading. I've been a committed fan of TWiV for a couple of years now. My older brother introduced me to your podcast. He is an assistant professor at Boston University and during his PhD at Columbia attended an immunology course taught by you. Since being introduced to TWiV, I have been spreading the word and now have many colleagues listening to the podcast. Thanks to you and all your co-hosts for the excellent work you do each week to provide us with current and relevant information. I should also mention I just purchased a year's subscription to CuriosityStream and enjoy watching these information programs with my kids. Big hello from all your Canadian TWiV listeners. Current temperature Celsius. Richard. All right. Cool. So let me just run quickly through some of these. I love them. I love to read them because these are a lot of people that don't usually write, and they tell us about themselves. I think it's really cool. So one from Aya. Was that how you say it? Aya? I apostrophe A-H? Iya? I forgot.
I think it was Iowa.
Anyway,
Aya.
He just said, boy, I hope I'm the 50th. Casey wrote, I'm writing in again. Am I the 50th? I noticed you did not draft the rules of entry, so he might submit multiple emails on multiple days, but he didn't. Second-year master's students in neurobiology at Vanderbilt. He's applying to PhD programs. He went to Cornell. He believes that we'd have a lot to talk about. He wants to ask a fun question. Do you guys and gals feel that in addition to your current subfield of research, you maintain other pet research interests? Although I study regulation of dopamine signaling, I always perk up when I hear mention of mitochondrial genetics. Love to hear what other subtopics intrigue you all. Thank you, Vincent, Dixon, Alan, Rich, and Kathy for hosting this podcast every week. It has quickly become my favorite, and I always devour the new episodes Sunday night and Monday morning at the bench. I'm also an avid listener of TWiM, TWiP, and TWiVo. I'm always pushing my grad student friends to listen in and often use the episodes to find new articles for my monthly journal club presentations. Recently, I presented synchronized cycles of bacterial lysis for in vivo delivery. Have a great week. I really hope I'm number 50. Sorry, Casey. Not everyone can be number 50, obviously. Pet research interests. Well, I consider all of virology, although I don't work on it all. But you don't really work on something else. You're just talking about something you're interested in, right? You perk up. I have to say, since I started doing TWiV and blogging, I'm interested in all virology. How about you, Kathy? Do you have a...
I always used to say if the virus I was most interested in, besides the one I work on, was influenza. And that's still probably true. And then areas outside of virology, I'm always interested in mouse genetics, things, particularly anything that's sort of new technology that makes mouse genetics work even better for virologists.
What about you, Rich?
Wow. Lots of things. I mean, it makes me think back on times when I was much younger, when I always had several projects going on at the same time, some not at all related to what I was doing, but that's a long story. In detail, in terms of a little thing, I've been interested in how in viruses, and I presume in cells, transcription is coupled to DNA replication. That is mechanistically really strange. But beyond that, there's a group at UF that works on triplet repeat diseases, and they're very good, and I always pick up when I hear about that. Huntington's, myotonic dystrophy, and et cetera. And I've always liked plants. Anything planty is good.
How about you, Alan?
Everything's a pet research interest for me.
Yeah, right.
That's one of the advantages of being a science journalist. I can have a new pet interest every week or every month, and it's wonderful.
All right, Hannah calls us the Five Stooges of TWiV. Here's a time not getting to the end of the podcast till Tuesday morning might work in my favor. She liked the blooper reel. Good to know we're all a little Skype challenged. Still loving your podcast from blustery Boulder, Colorado, where the gusty winds are blowing in, colder temperatures, and the first snow in the mountains. Amelia wanted to let us know that she loves the show. We're all doing a great job. Margaret, first-time writer, long-time fan. Her interest in TWiV podcast started with TWiV 4 years ago, but latched onto TWiV to stay current on viral developments. Currently working towards a master's in biology with a focus in acanthocephalin systematics at SUNY Oneonta. Thanks for putting out great edutainment and for encouraging newbies like me to write in with our bids for a chance to win a great book. Here you go. So she's never written before, and we get to know that in Oneonta, it's 10 degrees Celsius, partly cloudy, no wind. I remember Oneonta, not too far from Ithaca.
I've been through Oneonta.
Yeah. Christopher has been listening to TWiV for 2 years, started to listen to the other TWiV Microbe TV podcasts in the past since 6 months or so when he took a habit of taking an hour-long walk every day. That's very good. Another podcast I enjoy is People Behind the Science, which I've been on and a number of other people have been on as well. An episode of yours I listened to around a year ago discussed mechanical keyboards. That got me thinking about how I enjoy learning what kind of equipment and tools people use for their job. My question for you is, have you ever had a setback due to inadequate quality of office supplies? Currently 64F and 85% humidity. We have a lot of challenges with Skype. I have a lot of equipment for the podcast here, and that sometimes is a challenge. Something breaks. But of course, in the lab, things break all the time. I've just spent the last 2 days trying to fix a computer that was on the fritz. I just got it working again. Any other stories?
Man,
it's the opposite for office supplies. Ever since you picked jet pens, I have no problem with pens because I just throw out or give away anything that isn't a jet pen. And I just got a new set of pens last night and more refills. So I'm very happy.
Pens are a great way. You should go to Japan, man, because that's where most of the jet pens come from. They have so many cool pens.
Yeah.
I went crazy. I bought like 15 of them. What about you, Rich?
Well, what jumped into my mind when he mentioned that first thing, although it's a little off topic, is when I was a diehard Mac guy for many years.
Yeah, now you went the dark side.
The University of Florida beat it out of me because when I got here, being a Mac person in what was essentially a PC world was just impossible. Yep. And so I spent all of my time doing file conversions and stuff, and I finally gave up.
Ellen, you don't have any mechanical problems.
I had a MacBook fail catastrophically as soon as I showed up for a conference, like a 3-day conference that I was covering. Total massive disaster, and I ended up taking notes by hand at first, and then luckily, yeah, I know how horrible that was. But luckily, I found that the conference center was next door to a mall that had an Apple store, and I went over and they confirmed that they couldn't fix the MacBook right then, and I bought a Bluetooth keyboard and took notes on my phone.
Wow. And now you carry around an iPad, right?
Now I use an iPad, which the phone worked surprisingly well. But yeah, now I do that kind of a setup with an iPad.
One thing I've done on multiple trips, I think 3, I forget my computer charger.
Oh, yes.
And then I have to buy one in the airport or locally. I remember at ASV in Canada a couple of years ago, I got there and I said, I don't have a charger. So I got in the taxi, I found the local Apple store and I told the driver, I said, bring me there, wait for me, and then bring me to the hotel.
I bet at this point you could go to any hotel lost and found and
you're right.
Yes.
get a charger and an umbrella, probably.
Yeah.
Yes.
Paul says he loves viruses, but not so much the ones that make me sick or break my computer or increase the risk of cancer or increase the pathology of other viruses. Perhaps it's the knowledge of biological systems from the organelle to the ecosystem provided by viruses that I love. That's pretty good.
Yeah.
Casey really liked the PIC 538's Science Isn't Broken article.
Yeah.
Jorit, Masters of the Twix Universe. I hope to still have a shot at winning, although I have only finished listening to this week's episode Thursday as I listened to TWiV on my cycle commute to the Academic Medical Center in Amsterdam, where I work as a resident in clinical microbiology, virology, and clinical parasitology. My PhD work was on respiratory tract infections and acute respiratory distress syndrome in ICU patients. I've done a postdoc with Professor Menno de Jong in Amsterdam, or maybe Jong in Amsterdam, right? In 2011, I was struggling to find my path in medicine, and discovering TWiV and later TWiP and TWIM has helped me discover my passion, viruses, parasites, bacteria, and helped me realize I had to pursue a career in clinical infectious diseases. Although I'm currently spending all my time practicing clinical medicine, I hope to be able to take some time during my residency to get back into science, and would love to keep doing both patient work and science throughout my career, and hopefully even set up my own lab. Cool. Brian tells us we are all wonderful. Chaim says, I'm more of an immunologist, but it's been great for me. I study host pathogen interactions, so being exposed to more virology on TWiV and TWiVo for almost 2 years, I always enjoy it. Eric is Eric Carter, who made us, Dixon and I, the books. Thrilled to hear
all right.
Thrilled to hear us read his letter on that episode. The book you're giving away appears to have some really spectacular and beautiful images of viruses. I'll probably buy one even if I'm not number 50. I guess that's the logic behind the promotion, right?
Yes.
Yeah, David, thank you much for your weekly episodes that keep us educated. Dorota would like to point that writing today is not my strategy to win a book. I just happened to listen to the last TWiV podcast about Serink and NAF on my way to the uni and decided I should drop you an email while waiting for my Western blot. Working on Serink project during my PhD at the Institute of Molecular Virology in Ulm, Germany. I've been waiting for you to discuss these two papers for about a year and started losing hope, thinking that this topic might be missed due to all the Zika hype. It was really great to hear that you found this discovery of a new restriction factor as interesting as I did and decided to share it with your listeners. Special thanks to Jeremy, who did a great job explaining not only the papers, but also the background story of people involved in this discovery. I really hope Serink will continue getting the attention it deserves, and scientists, including me, will figure out what this enigmatic protein does, apart from decreasing virus infectivity, why it's so conserved, what other viruses can it restrict, what is the exact mechanism, etc. More questions and answers at the moment. Keep tweeting. I really enjoy listening to your wonderful stories about virology, parasitology, and microbiology. Best wishes from wet and gloomy Ulm, 9 degrees Celsius. Someone else take a few in rapid order. Alan.
Sure. Richard says, Dear Brahmins of TWiV, please collect a bacterium from deep underground, isolate a naturally occurring phage, splice it into that phage, a coded apology to future generations for our selfish failure to prevent climate change, and inoculate the original rock stratum with bacteria carrying the message. And suggests that actions taken and sacrifices made to mitigate the harm are good deeds to be proud of. So Sam says it's 73 and sunny in Tucson. Forecast is 92 with 61% humidity. Fall is officially here.
Oh, right.
Okay.
It's gone below 100.
Yes. James says, James is a first-year medical student from University of Minnesota in Duluth, and much more self-conscious in my habit of addressing unisex groups of people as you guys since listening to TWiV. Spent a year in Louisiana last year and realized I'm not a huge fan of y'all, though I will use it sometimes, and is going to switch to friends, but maybe in professional environment, colleagues. And says, listening to TWiV is great for me because I'm very interested in the virology and lab science. But as a medical student, I'm thinking about whether I'd like to specialize in infectious disease or public health, and wanted to know if we're interested in interviewing more infectious disease practitioners, or could refer him to older episodes that feature some. Didn't start listening to TWiV until after episode 300. Recall searching once or twice and coming across an MD PhD student interview episode, one with a researcher in Brazil who was a physician, and Henderson's interview, but wanted more suggestions of clinicians. And I think we have some of those, and Vincent, you've put in...
check out TWiP, where we have a resident infectious disease physician on the show.
Well, you have an infectious disease physician on the show. He's not a resident.
A resident, yeah. He's not, certainly not. He's an experienced fellow, yeah. But I think we've done more on TWiV.
Is Jeremy an MD?
He is an MD, although he doesn't practice. So
he doesn't act like one, but
I mean, the thing I asked, a long time ago, someone said get a doctor on the show, and he volunteered. He said, but I don't really see patients, so
right. He's a researcher, and we've had Ian Lipkin on a number of times, but he's we primarily talk about his lab work. But yeah, we've had...
Scott Hammer was on a long time ago.
Yeah, Scott Hammer.
And
We've
then,
had a few people.
the one from Vanderbilt had an MD on that one as well. They're scattered all throughout there. We're not so bad with ID physicians.
Yeah.
All right.
And then,
the lady from University of Michigan who works on HIV. She's a doc. What's her name?
Yep, Kathy Collins.
Kathy Collins.
Okay.
And you read. I'll look through the list.
all right. Tubby writes, I'm sure you guys have long since passed the 50th email, but figured I'd send one anyway. I much enjoy the shows, though a virology book is probably wasted on me. Be nice to Dixon.
I love that one. That can't be wasted on you. Learn something, right?
No, this looks like a
Especially
lovely
this book has
virology
such
book.
cool pictures and stuff. Yes. It's really good.
Jarrett writes, hello, TWiV team. Taking a break from cramming for my accelerated anatomy and physiology course. Hopefully your physiology doesn't get too accelerated. In the event I'm not winning, I would still like to express how pleased I am Rich Condit is moving to Austin, where I live. I hope this increases the odds of a live TWiV event here in the future, as that would be a real treat. Weather here is beyond reproach at 27 degrees Celsius, with only a few cirrus clouds up in an intensely blue sky, the enjoyment of which has been precluded by my obligation to study.
I think we ought to have a TWiV listeners regular luncheon in Austin.
There you go.
So,
we
I've
can
already
go.
I've already connected with Neva.
We can go. We can travel there, especially. Yeah. We could do 500 there if you want. Go to barbecue.
Yeah.
Boy, I know an insane barbecue. It's
well, as we said before, there's lots of music venues there, and we could do 500 there. That's fine. Yeah. Where are you? You finished Jarrett, right?
Yeah, so we're up to Jolene.
Jolene, Kathy, you want to?
Sure. Jolene, again, doesn't think she's going to win the book, but she's entering anyway. She found out about Dr. Roosink in 2014 and immediately saved a PubMed search on her titled Good Viruses Lady. So, yes, she talked about the kind of viruses that don't make you sick. Yeah. And so she talks about several aspects of that. This is technically my second time writing in because I entered the second book contest, but I came in 6 minutes late, and evidently she was late for this one too. This past summer, I was fortunate to meet Kathy and Vincent for the first time at ASV at the live TWiV, which was surreal to attend. I had previously met Rich over lunch when he visited IUB to give a research seminar, and I really enjoyed talking to him then. I listened to most episodes at 2x speed, though, so in person it seems like you all speak much slower. Anyway, she'd like to have the TWiV team visit IUB to do a TWiV there with as many hosts as possible. Adam Zlotnick has invited Vincent, and I want to remind him that if he hasn't yet scheduled that visit, we still want him to come. She's willing to organize the grad students. And she goes on to say that she did a gift subscription to Curiosity Stream to her brother to support the efforts. She doesn't mind the ads during the podcast. And she has a listener pick. I don't know if we're saving that for later. Yeah, we could save that for later.
Can you copy it? I'll copy since you're still talking. Go ahead.
And anyway, so, and maybe we'll just do this whole thing when we do the pick.
Yeah.
That sounds good. Yeah.
Sure.
Okay. And then the next one is from Falai. Hello, folks. Currently listening to TWiV 410 in Nigeria. I've been doing this since 2010, I think. You folks are doing a great thing educating the world about viruses. Keep up the good work. Cheers. Heidi writes, Dear TWiV team, I hope I'm neither too early nor too late to have a chance for winning the book of Marilyn Roosink. And when you'll send to us and when you send to Australia, then I'm looking forward that you would send to Cambodia as well. Because another listener last week was worried that you wouldn't send it to Australia, but you only have to send it to Canada, evidently, for this one. She enjoyed episode 409 because it was reminiscent of the vivid discussion with her PhD supervisor about the importance of NEF for her lentivirus-based pseudoparticle system. So she really enjoyed the history behind the best-studied proteins of all time. As a researcher highly involved with dengue and now Zika, I had special fun with the recent episode 410. The discussion demonstrated perfectly that there are no easy solutions for dengue or a Zika vaccine. I have a comment to Marianne's email. There is no black and white in the maturation process, meaning that there are not only complete mature and immature particles secreted from infected cells, but also particles in intermediate states. And she gives a reference to a 2010 Journal of Virology paper. And to make the situation even more complicated, the conformation of the dengue conformation of the dengue envelope proteins is dynamic, which changes the epitope susceptibility. And of course, as always for dengue, there are serotype-specific differences. And she cites a Richard Kuhn paper from 2015. Current weather here in Phnom Penh, no rain, what is really rare during the rainy season, 30 degrees Celsius outside and 16 Celsius in the lab, because our great Khmer technicians like it cool, 70% humidity. Thanks for the brilliant weekly entertainment. Best, Heidi. P.S. I might have to read another Richard Preston book then, The Hot Zone. Near writes, hi all, I hope I'm number 50. And then there's a little emoticon that looks like a yellow gumdrop with a face on it. My name is Near, and I'm a postdoc at the University of Chicago. I just moved a month ago from Israel. I've known about this podcast for years, but I never found the time to hear full episodes. Now my hour-and-a-half commute each morning and evening is a perfect chance to catch up. Keep up. Yeah, it's a long commute.
That's quite a commute, yeah.
Rohan writes, hey ho, TWiVers. I hope this is the 50th email you've gotten as part of the contest. If not, then I guess there's always the 4th copy of the book to look out for. I've been listening to TWiV since I enrolled for my master's program in molecular virology in 2009, and I should say it has helped me a lot in understanding the basics of virology. I love the way you discuss papers in a simple manner, which enables anyone with an interest in the subject to easily understand what you're talking about. I'm currently pursuing my PhD in pharmacy from Cardiff University in the UK, and I'm working on a host-targeting antiviral compound active against measles and vaccinia viruses. Best regards, Rowan. Okay, Angus writes, hi all, I hope I'm number 50. I'm an undergraduate student in my final year studying applied pharmacology at Queen Margaret University in Edinburgh, Scotland. It's just turned autumn here, the trees are becoming amber in color, and the temperature rarely gets above 15 degrees Celsius. I recently discovered your podcast after trying to find a comprehensive lecture on the background of Ebola and HIV. Lo and behold, I found TWiV 408 and thoroughly enjoyed listening to David Quammen's stories of his time in the field. I've since subscribed to both TWiV and TWIM, and I've enjoyed listening to all the episodes released since. Ruslan writes, hi team, love your work. Robin writes, great show. Justin writes, hello TWiV folks. I primarily listen to Twix while I'm driving, so I consistently forget that I mean to email in by the time I reach my destination. That or while I'm doing bench work and have my hands in gloves. I thought for sure I would have missed this one, but after a week went by without hitting 50, I thought it must be worth a shot. Here's hoping. Best, Justin. Sorry, Justin.
I wanted to point out that one of them, Robin, is from Missoula, Montana. So we're finding out where Justin's from. SUNY Albany. And why don't you take one more?
Sure.
Kathy?
Alex writes, hi, Kathy and fellows. Greetings from Boulder, Colorado. It's a beautiful fall evening here, 61 Fahrenheit, which means 16 Celsius. I'm listening to episode 410 while making some lasagna for the week. I've enjoyed listening to your discussions throughout my graduate school career, and I believe it has enriched both my understanding and enthusiasm for my work. Keep up the great work and be nice to Dixon. Best, Alex.
All right, Rich, why don't you end with the last few?
So is this Nellie or Neil? N-E-L-E. I don't know. Nell. Thank you very much for giving people abroad a chance as well. I hope you'll get a 50 one day. Still dark outside, 9 degrees Celsius. Greetings from Rotterdam. Lyson or Listen writes, I love viruses. I really do. Michael writes, I briefly contemplated sending in 50 emails last week to guarantee a win because you did not specify a limit to the number of entries per person. I meant to send it later in the week, but never quite got around to it. But since you mentioned you hadn't reached 50, I figured now is as good a time as ever, so here it is. And as an aside, I wanted to ask if Dr. Condit is related to Ira Condit of fig breeding fame. Wow. Not to my knowledge, though. I think all Condits share some sequence back there somewhere. I love all the shows and wish to convey my gratitude to all the hosts. And I got a copy of People, Parasites, and Plowshares for Christmas a couple of years ago, and was more giddy than my 2 daughters were. Most sincerely, Michael. Mary Ann writes, listening to the podcast while taking a walk in the autumn sun in Halifax, Nova Scotia. Our province got quite a wallop from the remnants of Matthew. Some areas got over 200 millimeters of rain, tons of flooding. I would love to win the book. Cheers, Mary Ann. Blair writes, "Hi, Vincent. Though I would like to, thought I would try my luck at the competition. My name is Blair Smith. I'm 35. I'm a 35-year-old guy of Melbourne, Australia. Never written before, but I'm one of those dedicated listeners that hangs out for every TWiV, and TWiP especially. The rapport that Dixon, Daniel, and yourself have always makes me laugh. However, I find that TWiV goes way over my head. While I'm not a scientist, I do have a big connection to viruses as a happy, comfortable host for HIV and CMV, and I'm sure numerous others. Ever since my diagnosis, I became fascinated by viruses and really enjoy learning as much as I can about them. As a complete layman, I find that the way you deliver TWiV keeps me fascinated. Anyways, I wanted to say that I really appreciate the work that you, Dixon, Daniel, Kathy, Rich, and Alan do, as you all have certainly fostered a passion in me. Cheers, Blair." What a nice letter. Peter writes, "I love viruses, the book kind, not the kind that make you sick." Page are okay, too. And just came at 2 p.m. today, Friday. Alex writes, "Hello, TWiVsters. I'm a graduate student at University of Washington, St. Louis. I've long used the Twix podcast to keep my ears and brain company while my hands do the pipetting. I tried to be strategic with my email timing, so here we go. Fingers crossed for number 50. Book or no book, I wanted to say that I really enjoy the podcast and appreciate the time and effort that goes into them. Thank you for all you do, and I look forward to many podcasts to come. All the best."
All right. I just think this is great that we hear from people and they talk, and they wouldn't normally write in, right, because they don't have a question or something. And so I love it. It really connects us more with our listeners. We have a great, great, great set of listeners. We really appreciate it. It's wonderful.
Phnom Penh, Uganda.
It's all over the
Rotterdam.
world.
It's Amsterdam. It's wonderful.
Nigeria, yeah.
Wanted to say that Martin, who often writes in about not believing vaccines or certain viruses, he even wrote in and wanted to, maybe we should send him a copy. Okay, one last bit of info. I've just revamped the Microbe TV Cafe Press store. I've put all the shows in, cafepress.com/TWiV, so you can get mugs, water bottles, T-shirts, hoodies, zip hoodies, tote bags, even mouse pads, okay? And a buck from each purchase goes to helping us support the show. So if you want to show your love, and those of us who have supported us on Patreon at certain levels get a mug or a T-shirt as well. So if anything, just check it out, because I spent a lot of hours making it look cool, and I think it's pretty neat with all the logos. Let's do a quick Zika sit rep. Now, I looked at our Department of Florida Health daily update. 5 new travel-related cases today, with 1 in Brevard, 1 in Duval, 1 in Hillsborough, 1 in Orange, and 1 in Palm Beach. 6 new non-travel-related cases linked to Miami-Dade. 1 had exposure in Miami Beach. And the investigating is going on in the other cases. Now, there are a couple of articles that we found, one on CNN and one in the New York Times, about a new Miami neighborhood where they found. I don't know if this report includes those or not. Does anyone have a sense for that?
The most recent report on the Zika update is from 2 days ago. So if this is really recent, it would not include those. And the really cool thing, I don't want to interrupt the stream here, but this October 12th report, if you scroll down on it, has these wonderful timelines.
Yes, those are great.
Did you look at those?
They heard us talking last time, remember?
Man, they're really cool.
We said what I really like. Timeline, yeah.
What I really like is that, on the timeline, there's a whole bunch of cases a gaggle of cases of Zika. And then down below, there's what looks like a war going on with trucks and airplanes. Spraying repellent all over the place, and the cases go away.
Yep.
And whether that's cause and effect, I don't know. But it's the timelines are brilliant.
Yeah,
they're really brilliant.
These are really nicely designed, yes.
Yeah.
In one of these articles, they mentioned the fact that the hurricane seems to have dispersed mosquitoes in some areas. I don't know how they would conclude that. Maybe they're not collecting as many as they did before. But we mentioned that last time with Scott and Sharon, and we'll see the effect of that.
Yes.
All right, we have a snippet in a regular paper today. The snippet was actually sent in by our friend Anthony, who is over here in New Jersey and often writes, likes to listen. and it's a PLOS One paper called Presence of Vaccine-Derived Newcastle Disease Viruses in Wild Birds. It says many authors. First author is Andrea Ayala. The last author is Claudio Alfonso. It's from the University of Georgia, USDA.
SCPRL, Southeast Research Poultry Lab, Poultry Research Lab, also in Athens.
And then someplace in Ukraine, the National Scientific Center Institute of Experimental and Clinical Veterinary Medicine, São Paulo, University of São Paulo.
Yeah, 4 places in Brazil, Ukraine, Bulgaria,
and
and
the reason
Georgia.
is that they sampled wild birds from all these places. So Newcastle disease, we haven't really talked about here, I think, on TWiV. Maybe we have, but it may have been a long time ago. It's a virus of birds. It's a big problem for the poultry industry. Spread through direct contact between birds. This Newcastle disease virus is a paramyxovirus, a negative-stranded, enveloped virus in the same family as, say, measles virus. And this is spread between birds by body contact, discharges, and, if you have a lot of chickens, commercially raised chickens, this is a big issue because it spreads through them like crazy. And there have been outbreaks where millions of chickens have to be culled because you can't sell these chickens. Infected birds can have mild signs, respiratory signs. They can be depressed. They can have tremors, drooping wings, diarrhea, thin-shelled eggs, reduced egg production. But there are more serious cases where you have sudden death. One of the symptoms, including nervous symptoms, are twisted heads. And I found a picture of a poor chicken that shows this with lying on the ground. His head is completely twisted around. I feel so badly for this chicken.
Yeah.
I know his prognosis wasn't good either anyway, even if he's healthy, but because he's going to be slaughtered. He's a broiler. But it's sad. And so this is a disease against which we vaccinate in the poultry industry to prevent outbreaks that would compromise the sale of these birds. And this paper essentially finds that the vaccine strains that we use to vaccinate, chickens in commercial facilities can be found in wild birds.
And that's because they're live vaccines. They're not inactivated viruses.
Right.
Yeah, and they're very effective. They're easy to administer, and apparently they're very effective. They give good immunity.
Do they vaccinate every chicken?
Everyone.
My
You could do
God.
that. You could get that job, Rich, if you want.
My God, no, I don't want that job.
Yeah, they do. And they do the same thing with fish. They vaccinate every fish.
My God.
Well, in certainly in the chicken industry, I'm not as familiar with that aquaculture, but the industry itself is so thoroughly consolidated. There are really only a couple of companies running chicken farming in the U.S., and I don't know how it is in other countries, but here it's Tyson and Purdue and maybe a couple of other producers, and they contract everything out, but everything is done exactly according to what the producers want. And so all they have to do is say, you vaccinate the chicken, and the chickens will all be vaccinated.
Can you imagine how much vaccine that is? I was just enormous.
Tons.
It's a lot of vaccine, and that's a big industry itself, yeah.
Yeah.
Well, there are multiple ones. There are a couple, 3, 4, and they're all, as Kathy said, infectious, attenuated vaccines. They're very good, easy to administer. They give good immunity. So they're used as opposed to inactivated vaccines. And, of course, there's the possibility that we know the virus is shed from immunized animals, so there's a possibility that it could get into any wild birds that happen to be around. So they wanted to look in this paper if they could find vaccine strains in wild birds. So they did sampling at multiple sites, including all those places that we mentioned in the beginning, Georgia and the Ukraine and Brazil. And then they get samples from these birds. Oh, what kind of samples did they get? I forgot to look at that. Does anybody remember?
They got blood and cloacal and some other kind of swab. I wrote it down too.
Throat swabs or something like that.
Oral, yeah. Oral cloacal, yeah.
And then they basically sequence these samples to see what's in them. And they find, of course, viruses in a variety of birds in Brazil, Bulgaria, Ukraine, and the USA, mainly in columbiforms, which include pigeons and doves. Not Allen doves. And anseriforms, waterfowl, including ducks, geese, swans, and screamers. Anybody know what a screamer is?
I don't know what a screamer is.
That's a great name for a bird, a screamer. I guess
No,
scream.
but I had to look up dabbling ducks because that's another kind where they infect. And those are birds that tip down to reach aquatic plants and invertebrates below the surface.
You know, that happens.
They fly by springing into the air from the water.
They happen to be also a reservoir of influenza. I remember dabbling ducks. That is as well. So they can sequence these isolates and see that they're vaccine-derived. And again, these are from wild birds, and the isolates are very close to the vaccine strains, a couple of different vaccine strains, not just one. And they get them from all these different locations. 17 different species from 10 avian orders have this virus. So presumably what's happening is that somehow, well, we know the inoculated chickens are shedding virus, and somehow it's infecting wild birds. Now, I don't know how exactly that happens. I mean, I guess sometimes the coops are open and the wild birds can get in, but I've seen these huge production farms. It doesn't look like any birds could get in, but maybe there's some cases where that can happen. Or, of course, if they take out stuff from the floor of the chicken coops and throw it outside, then, of course, birds could pick that up as well.
Right.
A lot of their sampling was like 72 pigeons in Atlanta. Okay, so where the heck? I mean, I don't know what the range of a pigeon is.
Well, I think that was just a sample of convenience.
Yeah, right. But where did the pigeons get?
Yeah, where'd they get it from?
Tons of poultry farms in Georgia.
Lots of farms in Georgia, even probably close to the Atlanta area.
Yeah.
So I wonder if one thing they comment on here is that they don't think that the virus is being passed from bird to bird in the wild, but that these are all, did I understand this correctly, that these are all primary infections?
Yeah, because there's very little genetic diversification. It's over an 18-year period, and there's very little difference as opposed to wild strains, which vary quite a bit. Yeah, so probably.
So the pigeons going out and visiting the poultry farm?
Apparently.
Hey, there's good feed there.
Good point. And there was like 9 out of 72 pigeons, so that's like 10%.
And they say this is probably an underestimate because they only did. This is interesting. They only did a convenient sampling. You know, the specimens they could get. We utilized a convenient sample to make a causal inference, and they say we have to look more. They only had 54 isolates in total, but
that's a pretty big number.
Yeah, they're probably more widespread than this. Yeah,
and if they went to an area that's really heavily planted in chicken farms, you might find a higher rate.
So I found a site called Pigeon Facts. It's full of pigeon facts, more than we can go into. But homing pigeons have been known to fly 700 miles in a day.
Yes.
Wow.
So then I went and tried to be more specific, and I got to the Cornell Lab of Ornithology page for rock pigeons. But so far, I haven't found their flight range.
Is that do they play rock, scissors, paper?
I don't know, but they extend all the way up through Juneau, Alaska, and the southern part of Alaska, down to the very tip of South America in their range. So quite broad range. For
they call this reverse spillover. So I think David Quammen would like that, right?
Kind of like reverse genetics.
Yeah, right.
At least you didn't say
poison a term.
At least you didn't say a factor. Anyway, I thought this was interesting. There's no indication that it's a problem, right?
No.
It doesn't seem to be causing disease, although maybe they haven't really done the right study to look at that. But
there's no shortage of pigeons.
No.
Might be good for them.
Yeah. If it hasn't reverted, I mean, that's a study that could be done. You could take these isolates and inoculate them into the proper test animals and see if they have acquired some virulence. That would be interesting, right?
Sure.
Now, remember, we did a paper a long time ago about herpes virus vaccines against Marek disease for chickens, right? Mm-hmm. And they recombined with each other to produce a more virulent virus. Do you remember that?
Mm-hmm.
Right. Yep.
So there's no indication that that's what's going on here, but since there are multiple NDV vaccines, I guess it's a possibility.
I don't know with what frequency paramyxoviruses recombine. I'll look it up.
Yeah, it may be low. Yeah. Maybe it doesn't happen at all. It's a good point.
Certainly less than a DNA virus. RNA virus as well. That's probably an overgeneralization. But I think in a general sense, DNA viruses recombine more frequently than RNA viruses. And some RNA viruses do it pretty efficiently, and some don't do it hardly at all.
I think the plus-stranded RNA viruses are pretty good at it. I don't know about the negative. I mean, there's certainly reports in the literature, but I've also heard people say that it's very low frequency because the genome is covered with protein, and et cetera, but I'm not really sure. Why don't you look it up?
I'm looking it up.
Well, I tell people about our sponsor today, CuriosityStream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles, founded by John Hendricks, who was involved with Discovery Channel. So that means you're going to get real science, not reality TV. You can get it on many different platforms. Just a web browser. You go to the website, you can play the videos there, or you can use any of these devices that you use to hook up your TV, like the Apple TV, the Kindle, Amazon Fire, Chromecast, etc. And they have a wide variety of science, technology, nature, history. They have interviews. They have lectures. So I have their website open right now, and I just went to the search box and searched science. I got 97 different shows, and these are sometimes 2 or 3 episodes. Sometimes there's more. For example, the health of our oceans. A marine biologist, Sylvia Earle, reveals why threats of pollution and overfishing could have an impact on the oceans. We have Stephen Hawking's Universe, exploring quantum history with Brian Greene, my colleague here at Columbia. The Great Australian Fly, Waiting for Elephants. There's lots of cool stuff. They have virus stuff too. They have space. Here's one that I saw earlier. Order and Disorder. Jim Al-Khalili reveals how humanity discovered the rules that drive the universe and mastered 2 crucial concepts, energy and information. Lots of neat stuff here. You might like it. And the cool part is they have monthly and annual plans available. They start at just $2.99 a month, which is not a lot of money, less than a cup of coffee. And they also have a lot of super high-definition content, 4K. I have about 50 hours of that. And I just saw the other day an article about a new camera, which is 8K. Can you imagine?
Wow.
4K is hardly penetrated, and 8K cameras are already available. But nobody can.
I think the camera manufacturers are just getting desperate at this point.
To sell something new. Well, it may drive adoption. Who knows? Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 months free of one of the largest nonfiction 4K libraries around. Go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV. Someone asked in an email how long we're going to do this. Well, CuriosityStream is booked to the end of the year, which is great for us. You may get bored of it, but you got ways to get around that. And Drobo is through the end of October, so just a couple of weeks more. And I don't have anything lined up, so you may have ad-free TWiV again. Who knows? We'll see.
Yeah.
We will see. All right, our paper for today.
Wait.
Yes, sir.
Recombination among paramyxoviruses is rare.
Rare? Okay.
Rare. Okay.
Excellent. So it may not be an issue here.
May not be an issue.
So, the cool thing is with polio viruses, the oral polio vaccines, when you take them, you ingest them, they go into your intestines and they replicate and produce immunity, of course. But as they replicate, they recombine with picornaviruses that are already in your gut. We all have some. And everything that you excrete is a recombinant. They preferentially replicate in your gut. It's a recombinant with an enterovirus that you have in your gut. How cool is that?
Cool.
Mm-hmm. And no one ever suspected it until, of course, we were able to sequence not many years ago. But Albert Sabin had no idea that these things were recombining when people took them, but they do. And maybe that's what makes it a good vaccine. The recombinants have a good fitness and they replicate better. All right. This paper is quite interesting. It's in Science. It's called Discovery of a Proteinaceous Cellular Receptor for a Norovirus. And let me just thank all the authors for not calling it a factor.
Mm-hmm.
They call it proteinaceous because it is a protein. That's great. This is an awesome paper that, I've been working on receptors for a long time, and this paper has everything you would want to prove that a molecule is a virus receptor. Most papers have one or two of the proofs, and I must say that our paper published many years ago, I think 89 for the poliovirus receptor, had just one or two. This has all of them. It's really cool. And the first author is Robert Orchard, Craig Weiland. These are co-first authors. Is that correct? Yes, they equally contributed. Mm-hmm. Then we have Dunch, Baldridge. Dench. Judy Dench, right. I should remember that.
Oh, John Dench, right.
No, but Judy is the one I would remember.
Right.
The actress. But yes, it's John. Baldridge, McCune, Lee, Pruitt, Miller, Nelson, Fremont, and Herbert Virgin is the PI here.
Also known as Skip.
Yes.
Yep, Skip Virgin. And this is all about murine noroviruses, which we've talked about recently because we had an interesting, first we talked about development of a B-cell culture system to propagate the viruses from Stephanie Karst and Christian Vobus, and then more recently the system developed by in Mary Estes' lab. These are for human and murine noroviruses. But this paper, they look at the receptor for murine noroviruses. These are non-envelope plus-stranded RNA viruses with a proteinaceous capsid. I'm not going to say a factor capsid, am I? No. Now, in mice, they can, depending on the strain of norovirus, can make a persistent or an acute infection. And they want to know what is the receptor needed to get these viruses into cells. The approach is...
And you do this in mice because you don't have a robust culture system for the human viruses. So this you can grow a ton of virus, you can do plaque assays, you can infect the mice, you can do whatever you want.
Right.
It turns out, because I talked to Craig, the second of the two first authors, and I had email exchange with Rob, the first author, that wasn't exactly what they set out to do when they started doing their CRISPR screen. Skip wanted to find non-canonical autophagy genes that are involved in control of pathogens.
Yeah. Well, what they do is a CRISPR screen, of course, where they take guide RNAs from all the open reading frames in the mouse genome, and they deliver them into cells. They choose a macrophage-like cell called BV2. They've already put Cas9 nuclease in these cells, and they deliver the guide RNAs by lentiviral vectors. The idea is the guide RNAs will guide the nuclease to specific genes. They will cut and make a deletion, and when it gets repaired, you now have a null in that gene, and you could take a whole library of cells disrupted in this way and then infect with virus and ask what survives.
So you have all these individual lines, each missing hopefully one gene or gene product, and then you're going to look at what's infectable and what's not.
You do it as a pool, of course, and you just take the cells that grow after infection. You can pick them and take DNA out of them, and then amplify DNA at the site of disruption using primers, and then you can sequence it and say what genes, when disrupted, allow cells to survive after norovirus infection. So they get
and this is what they describe as an unbiased forward genetic approach.
Unbiased forward, yes. So they have a bunch of genes, and they end up looking at cell surface molecules, because if you're looking for a receptor, that's what you would look for. But I guess, Kathy, that they were initially interested in non-receptor genes, and that's why they did this screen, right?
Right, but they also thought they would get this out of it, too.
So they do get other genes involved in replication, but the focus of this paper is the receptor. So the molecule, the gene that they focus on is called CD300LF, and encodes a cell surface immunoglobulin domain containing protein. Now, we identified the polio receptor in 1989. It's an Ig-like protein. The same year, the rhinovirus receptor was identified as an Ig-like protein. And a few years before, the HIV receptor had been identified as an Ig-like protein. So it's very interesting that many years after all that, here's another one. So
these Ig-like proteins are all over the place. That is a theme that is used over and over and over again.
Right. Of course, now there are non-Ig proteins in the cell that serve as virus receptors, but a lot of them are Ig-like, and that's probably because these are adapted to specifically interact with other proteins, right? They have the Ig domain, which is binding antigen, for example. So I guess it's not surprising.
Okay.
So they make a disruption. They make two independent disruptions of this gene, CD300LF, and they show that it blocks virus, the ability of both norovirus strains, the acute or the persistent strain, to replicate. And they put in these disrupted cells, they transfect viral RNA in, and you get virus produced, showing that disrupting CD300LF blocks entry. That's a beautiful experiment right there. They have an antibody against CD300LF. They show that this blocks virus infection. They produce a soluble ectodomain of CD300LF. That is, you cut off the transmembrane domain, you produce the protein in cells, it's secreted, you purify it, it blocks virus infection in multiple cell types. They look at binding studies. They can show in a binding assay that norovirus binds to these BV2 cells. The cells in which the gene is deleted, the virus doesn't bind to them. You can block binding with soluble CD300LF. You can block binding with antibody to the protein. So that is all. And they even make a mouse lacking this gene and show that they're resistant to infection. I mean, these are like, most papers only do one or two of these things, and they say, we've identified the receptor for the virus. This does it all. This is really, really remarkable. Now, one thing that's very interesting, previously it's been suggested that carbohydrates might be involved in both murine and human norovirus infection, right? So in humans, there's this association between, certain blood group substance classes and infection. They make a mouse deficient in an enzyme called FUT2, which controls histoblood group antigen status, and those mice make as much virus as wild-type mice. And if they treat cells with an enzyme that reduces cell surface carbohydrates, doesn't alter binding. So they say that looks like proteinaceous receptor is really the main thing for getting these viruses into cells.
There's another paper that came out from a Japanese group just recently, and it was submitted a little bit later than this one. They also found the same thing, so it reminded me of Jeremy Lubin saying, it's nice when something comes out and it finds exactly the same thing. And in that lab, they used a different macrophage cell line, the raw cells. Mm-hmm. They used a different CRISPR library, but they came up with, they did 2 screens, and they came up with the CD300LF, and in the additional genes that they find, as being important, none of them overlap, at least the ones that are found in Figure 1A of the Orchard and Weiland paper. I didn't go back and look at the complete list. Maybe some of the others overlap. But in that paper, they do find a little bit more importance, or they find some, they confirm the importance of carbohydrate binding, although they say that it's much less than the importance of the protein factor. And that's done, again, in a different cell line. So that could be part of the explanation. But when I talked to Craig, he said, it seems pretty clear that the protein receptor is the major thing. It causes a 2 log difference, whereas the carbohydrate elimination causes a 2-fold difference.
So have they looked for the human CD300LF, whether it binds human noroviruses? Did they say anything about that?
I didn't ask that.
Yeah, because
it's not mentioned in either paper that I saw.
Right.
I'm sure they've done it or they're doing it. One other cool experiment. Well, actually, there are many more cool experiments here.
Another one. Huge amount of work in this paper.
It is. It really is.
I have to tell you how that came about, but go ahead. HeLa
cells can't be infected with murine noros, but when they put the gene for CD300LF into HeLas, they can now be infected. So the only block between norovirus replication and HeLa cells, mouse norovirus, is this receptor. And it's so reminiscent of polio in mouse cells. It's the opposite. So mouse cells can't be infected with polio, but if you put the human polio receptor gene into them, it replicates. And that's the only block. One other thing I want to just end up with is that they determined the crystal structure of CD300LF, the ectodomain, at 1.6 angstroms resolution. And they could see typical immunoglobulin-like folds. Now, the first Ig-like domain seems to be the one that's interacting with virus. And this is reminiscent for me because we had the same kind of work we did with polio years ago. Ig-like domains have a very specific fold. And the first domain has two beta strands called the C, which form the C prime loop, which is very important for binding of the receptor to polio. It's important for binding of antibodies to antigens. It's important for binding of HIV to its receptor CD4, and they do some mutagenesis, which implicates that loop in binding as well. And they also see from the structure that there's a metal there that can bind, and it seems to be involved in virus entry. So this is very interesting and reminiscent because years ago we published a paper where we took a human and a mouse receptor for polio and swapped bits and used that to map the binding site. And this is exactly what they're trying to do here. They do substitutions of human and murine CD300LF to try and map the binding site. So, folks, we did this in 1994 for the polio and the polio receptor. So you should go back to that because sometimes you can learn stuff from things that are published many years ago. Anyway, it's an awesome paper. I like it.
Very cool.
Yeah, so I wanted to tell you a little more, just tagging on to what you just said, because I saw you already commented on that in the show notes. I asked Craig, and he said, oh, yeah, we were definitely very aware of Vincent's paper. And Craig himself had done his PhD in Bob Dome's lab with HIV entry, so he was very familiar with those kinds of swap things. Couple things to tell you. Both of the first authors are postdocs. They're both going to be on the job market next fall, so watch out for them. In the case of Craig, he did his undergraduate work at WashU with Dan Goldberg working on malaria, but he decided the genome was too big. He had always known that he wanted to be a physician, but doing that research in the Goldberg lab, he fell in love with research, so he decided to go for both. So he's an MD, PhD. He went to Penn for his MD and PhD degrees, and then came back to WashU for his clinical pathology residency, and then did research in Skip's lab, and then is now just a postdoc in Skip's lab, not doing any clinical things. And the other first author is Rob. Let me pull up his email. He grew up in Texas, always knowing that he wanted to do basic microbiology. He worked on phage biology at Texas A&M, and then did his graduate work at Neil Alto's lab at UT Southwestern on the cell biology of secreted bacterial virulence proteins. And then he went to Skip's lab about 3 years ago to get more in vivo relevance and mouse model experiences. And he says, working with Craig has been awesome, and I'm not just saying that since he's CC'd on the email. We both have been highly motivated and use each other's strengths to really put this project together in a quick fashion. While we approach the science differently at times, our skill sets have been very complementary. I'm grateful to have been able to do science in a fun and collaborative way.
That's really great. Having a real partner doing science is just wonderful.
Yeah. So they started this in January of 2015, and they submitted it in December of 2015. And there were a couple of things that kind of helped along the way. So one thing was that, when they got their surviving clones that survived the mouse norovirus infection, and the thing to do then is to send that for deep sequencing. But they did some quick and dirty Sanger-type sequencing, I think is what he said, and 5 out of the 6 reads came up with this CD300LF. So, as he said, then we spent $10,000 more on the sequencing of all the rest of them and got those sequences. But shortly after that, getting those 5 out of 6 reads in the CD300LF, I guess Skip was talking to David Fremont, another person there in immunology and pathology, who had already crystallized CD300LF.
Eh.
And not published it. And they had milligram quantities of this in inclusion bodies sitting in the freezer. So within 24 hours, they had the soluble form that they could use in their inhibition assays, and the sequences, the crystal structure had already been solved.
Nice.
So that, and I said, did they have a bunch of CD300-like proteins or just the CD300-like F? And he said, no, just F.
Wow.
So that was pretty amazing. And then, again, just going back to this Japanese paper, I haven't had time to look at it in great detail, but they also cite in the supplemental data the secondary and tertiary structure of the molecule using a PDB file. So I just plugged in that PDB file, and that structure has David Fremont's name on it in the PDB database. So even if it hadn't been published, it just says to be published, it was there and available for the Japanese group. So that was kind of nice. So I asked, so when he told me about getting the 5 out of the 6 reads to be one protein, I said, so that was probably the most exciting day. He said, yeah, March 3rd, 2015. So that was pretty cool. I asked him a little bit, Vincent, you didn't mention the fact that they have some evidence that some other factor in serum contributes to this as a receptor, and it's something less than 5 kilodaltons. So I asked him if some biochemist was working on that, and he said, well, we're just kind of using a candidate gene or candidate molecule approach, and you can imagine candidates that are less than 5 kilodaltons. So they have some ideas about that. Let's see. It was just a really fun conversation.
They also made the knockout mouse.
Yeah. Oh, yeah.
And they have these one-liners in the paper like,
yes,
we generated knockout mice to test the in vivo role.
Right.
We determined the crystal structure. Well, now that's a little more obvious. But Mike,
right.
a year's work, and it's incredible.
Yeah, so I asked him, when did you start making the mouse? Well, that was June of 2015, and they had it by the end of January 2016. But they had submitted the paper in December, a month before they had the knockout mouse. So then when it came back, they put in the knockout mouse data, but they wanted to wait until they had mice from a heterozygous breeding strategies. Sorry. So they first got mice from a homozygous breeding strategy and tested that, but the reason they wanted to use mice from the heterozygous breeding strategy is because that would generate wild type mice from the same litters that would have the same microbiome. And the microbiome being important for the infection, that took another 4 months. And so that was pretty cool about that too. But yeah, I was like, they made a mouse, they crystallized and solved the structure. It was just amazing. He said there were already some knockout mice out there, but they figured it was faster to make it themselves because by the time they did MTAs and got the mice, and then they always re-derive everything within their barrier facility to be a mouse norovirus free. And by the time they did that, they could just as easily make the mice. So that's what they did.
Wow.
Yeah. Yeah, it's hard to get mice in from other places. It takes a long time. Here you have to quarantine them forever. Yeah.
Yeah.
All right. Very good. Thank you for that.
Mm-hmm.
Very cool.
I want to tell you about our sponsor of today's show, Drobo. Drobo makes storage solutions that you connect directly to your Mac or Windows or Linux box using Thunderbolt or USB 3. There are also network attached Drobos, and you use them to store your files. They come in different sizes, 5 drive bays, 8 drive bays, or 12 drive bays. And they're a little different from garden variety RAID. Someone wrote me a while ago saying, Drobo is RAID. Well, it is, but it's a variety of RAID that's a little bit different from normal RAID. Sometimes it's very difficult to recover data from normal RAID arrays. For example, Drobo's founder had a RAID array. He put his honeymoon pictures on it, and he lost them. And so he used that as an impetus to start the company that we now know as Drobo. Drobos protect your data against single or double drive failure because the data are replicated across all the drives, whether it be 5 or 8 or 12. If one of the drive fails, you simply take out the drive and put a new one in. You can tell if a drive fails, a red light goes on next to it. It lasts for a long time, for your lifetime. If you need more space, if you fill up the Drobo, you just pull out the drives and put in bigger ones, and the data will be reproduced across them. And they're very simple to use. They have lights on the front next to the drives. If they're green, there's plenty of space. If they're yellow, they're getting full. If they flash red, you need to get a new one because it's full. And if it's solid red, the drive is dead and you need to replace it. So really simple to use. You can attach them to your computer, or as I said, there are versions that you put in your network. You attach them to your switch or router. Very cool devices. I've been using them for years, so I'm really, really secure about recommending them for anyone. If you have data you need to protect in your lab, they're great for that. If you have stuff at home you need to protect, you can use that for that as well. So really good systems. Drobo likes to call them beyond RAID because they have advantages that typical RAID does not. Listeners of Microbe TV shows, including TWiV, can save $100 off their purchase of a Drobo 5D, 5DT, 5N, or any 8 or 12 drive system. Just go to drobostore.com and use the discount code Microbe100. You get $100 off of your purchase. We thank Drobo for their support of TWiV. Let's do a couple of emails, get into this backlog here. The first one is from Benjamin, who writes, "Hi, admin." It's funny. I really appreciate all your mails and am very grateful for it's been helpful. I don't know. I didn't send you any mails, Benjamin. You mean the ones we read? My question is this: what is the normal floral for viruses? Thanks, and I look forward to your reply. Is it cherry blossoms or magnolias?
Oh, you hate
I hope
them.
it's not
Couldn't
daffodils.
be them.
The normal flora. We've done a bunch of TWiVs on what viruses we all carry.
Yeah.
I don't think we know what the normal quote unquote flora is yet for viromes. People are still working on it. We all have lots of phages, of course, and we all have lots of eukaryotic viruses, but it hasn't been studied as much as the microbiome because there people have looked at different microbiomes in our body in health and in disease states. We know how they alter, but we don't really know that for viruses yet. Would everyone agree with that?
I think I would.
Yeah.
Right. We have some idea of what some of the viruses are in the baby's first virome.
Yep.
Based on that TWiV episode that we did, and there's some adult virome studies.
Yeah.
I think twins.
And there were the Shinola.
Yeah, that's right.
Episode that one looked at sewage viromes, which would be representative of human population viromes.
But, we have, as I said, we have lots of phages and we have lots of eukaryotic viruses. As I said earlier today, we have lots of picornaviruses in our guts that recombine with polio vaccine when you take it. So I think many people are working on this, and as the results come out, we will cover them. So that's what we do here. Mm-hmm. Alan, can you take the next one?
Sure. Jeffrey writes, "Doctors, I was looking for some information on whether or not we have found evidence of retroviral genes in mitochondrial DNA. I found at least one source that said it was unlikely, as it would require a retrovirus that could first slip into an animal and then slip into an effectively bacterial cell. In addition, I assume that the limited space available for mitochondria would make viable inserts difficult. However, I also found a reference to an endogenous retroviral integrase fragment, ERI-15, being involved in the destruction of sperm mitochondrial DNA. Unfortunately, that article, and gives a reference, is behind a paywall. My impression is that ERI-15 is provided by the cell and not the mitochondrion, but that abstract wasn't explicit. I also find it a bit coincidental that some antiretroviral drugs seem to have a deleterious effect on mitochondrial DNA," and gives a reference. Do you and or your co-hosts have a clearer answer to how retroviruses interact with mitochondrial DNA? Then there's the question of whether or not there are bacteriophages known to infect mitochondria or chloroplasts, shielded as they are by their greater symbiotic partners, bulks. I found a partial answer on Elio Schechter's Small Things Considered blog, and gives the link, but it only served to confuse me more. I'm sure Elio would be happy to hear that. Could you also provide some clarification on this point? The whole subject of the infection of endosymbionts would seem to present some interesting insights into the nature of these symbiotic relationships.
Interesting. I thought we talked about this before. Does anyone recall?
I feel like we've talked about phage and mitochondrial DNA. But
I mean, you can find phage-like sequences, and I think that's it's left over from when mitochondria were free-living, right? And they were infected with phages.
That sounds familiar, but I don't have the reference.
I don't think there's any evidence that retroviruses infect mitochondria. I think, as he says, I think these DNAs, these retroviral sequences come from the host. But what's interesting is I have a paper that I was going to do, that we shall do as a snippet next week. Stay tuned. It's really cool.
It's a teaser.
Yes, stay tuned. It's all about the whole subject of the infection of endosymbionts. Yeah. We're going to cover that paper, so stay tuned, Jeffrey. Kathy, can you take the next one?
Sure. Kevin writes, Dear TWiVologists, apologies in advance. I fell behind in my listening because of school, and now I'm rapidly catching up during the summer. In an earlier TWiV, I think TWiV 379, A Mouse Divided, Professor Racaniello mentioned a student named Isaac who held up a Be Nice to Dixon sign during his immunology guest lecture. I just wanted to set for the record that I was the person with the sign, so that when the future Be Nice to Dixon craze becomes popular, I can say I left my mark on history. Great show, and keep those episodes coming. It's currently 14C in Vancouver with sunny skies. Best, Kevin.
I know, Kevin. I thought that was you, Kevin. I mean, I don't think I said it was Isaac. But, okay, so Kevin was a student at Columbia, and he took my virology course. He took the immunology course when I gave this guest lecture. He held up the Be Nice to Dixon sign. I know it was you, Kevin. However, what I don't know is who was the fake Dixon on Twitter. You said, I think Kevin said it wasn't me.
Well, there are two fake Dixons on Twitter,
That's
right?
That's right. See, he is a cultural star, and therefore he gets fake people on Twitter.
Gets fake people on Twitter, yes.
None of us do. That's pretty cool. Rich, can you take the next one?
Brianne writes, Hi, TWiVers. I'm a longtime reader of Vincent's virology blog, but have only recently started listening to TWiV. Thank you so much for your discussions of preprint servers in TWiV 388. Before listening to your episode, I was aware of bioRxiv and knew of it as a preprint server, but I was not clear on the definition of preprint. I had originally understood that preprints were accepted unformatted manuscripts online, which would mean that they were being posted after peer review. Your thoughts are quite useful in explanation on this front. I have mixed feelings on the idea of publishing my work to a preprint server or even to open access journals. I feel strongly in favor of open access, particularly with regard to being able to see science that is supported by our tax dollars. As we are at a liberal arts college where we have relatively few subscriptions to discipline-specific journals, my students and I make extensive use of open access publications. However, as a faculty member at a primarily undergraduate institution, I worry about putting out unpublished data on a preprint server that others in faster-moving labs might scoop. Similarly, I worry about how open access publications and preprints will look as part of my tenure review. It's a shame that younger scientists need to make these sorts of calculations, as open access publications are helpful to so many audiences. Thanks for such an informative and entertaining podcast. I really enjoy listening and look forward to each new episode. I'm planning to incorporate TWiV, TWiM, TWiVo episodes in my courses, and I hope that my students enjoy them as much as I do. Count me among the listeners who would love to hear more immunology on TWiV, or even to have a This Week in Immunology podcast. Thanks again, Brianne. So I would say, I just have one particular thing to say here, that open access publications on your CV as part of your tenure review should be no issue at all.
That should be fun.
Some of the strongest scientific publishers now are open access, just PLOS pathogens as an example. So that's not an issue.
PLOS One may be a little more questionable just because people have mixed opinions about it. But in general, any of the other PLOS journals, if you publish in PLOS or PLOS pathogens or something like that, those are flagship journals that anybody would be happy to see publications in.
I took this differently. I took it that she was thinking of bioarchive as being the open access.
Yes, that is a different animal.
Yeah. So open access in those journals is one thing, but putting your stuff on bioarchive and how that might work on your CV or how that might work with Faster Labs scooping you, I can totally see.
I mean, having if you put a paper on bioarchive and then it's published in a good journal, that's fine for your tenure review. If you only put papers on bioarchive and never publish them anywhere else, then that could be a problem for your tenure review, right? So, but yeah, open access alone is not a problem at all. I would, Sharon and Scott last week, they're at an undergraduate institution. They put their paper on bioarchive, and they said because they wanted the data to be out there because it was, Zika. But I don't think they would do it for every study that they did. So,
right. In fact, I think Scott said that.
Yeah.
But they did appreciate the comments that they got from having it up on bioarchive.
Yep.
So
right. Yep. Yeah.
I think Sharon and Scott don't have a tenure review.
No, they don't.
Right.
They're in a hot field, obviously. So it kind of makes sense.
They don't have a tenure review, but they have a periodic review.
Absolutely.
For whether they're going to stay employed. So yeah.
Yeah. They told us all about that. Yeah. All right, and we do have audio immunity on Microbe TV if you want to check that out. It's a little infrequent, but they do make an effort. And
right, there's 24 episodes up there.
And someday we'll have a TWiV or something like that. It'll be a little bit. I have a number of new podcasts in my head, and I just have to find the time to, and the hosts. Hosts are important for podcasts, as everyone knows.
Especially microbiology podcasts.
Yes, you have to have the right microbiome.
The right host.
The right community.
Yes.
All right, let us do some picks of the week. Where are they? They're all the way down here. Wait, before I do that, let me just tell you about the 2017 ASM Scientific Writing and Publishing Online course. This is a webinar that explores the writing and publishing process with editors of ASM journals. It's an interactive series that'll cover titles, abstracts, figures, legends, text, of course, and the whole manuscript review process. The course takes place between January and April of 2017, and you have to register for it. There's a small fee associated with it. The registration deadline is December 1st. Learn more at bit.ly/SWPOC, Science Writing Publishing Online course. That's what those letters stand for. SWPOC 17. All right, Alan Dove, what is your pick this week?
My pick is another YouTube channel. I've been on kind of a kick of exploring science-themed YouTube channels. This one is Braincraft, and woman in the UK. She has an Australian accent.
She's in Australia.
She's now in Australia?
Well, that's what I got was that it was from Australia.
Oh, okay. Yeah, she a couple of the episodes she's in the UK, and I wasn't sure what her exact location is. Anyway, that's not terribly important. She's on the internet is where you can find her. And she does these videos on various scientific topics, primarily neuroscience, hence Braincraft. And the craft aspect of it is she does, to explain concepts that she's talking about, she does these handmade animations with paper cutouts and moving them around and using them to animate the concepts. And I think it works really, really well with a lot of what she does. And some of them are mostly her on camera, talking, if it's something that is fairly straightforward to explain, or some of them, she'll have footage of other things interspersed, but then a lot of them are heavily oriented toward this animation, which is a huge amount of work, and she does an extremely good job with it, and it's just really fun to watch.
Yeah, I watched the one on the blood-brain barrier. I thought it was really good.
Yeah. So you're trying to get ideas for a video series of your own, right?
I'm kind of kicking around some ideas, yes. So that's why I've been exploring YouTube channels. And, the drawback, of course, is, if I commit to something like this, it's going to have to be on a schedule, and I'm going to have to keep up with it, because so many of these things are easy to start but hard to continue.
Yeah.
And so I have a lot of respect for people who keep at it and keep releasing these very high quality science videos.
Well, if you decide to do it, we'd be happy to have you at Microbe TV.
I will keep it in mind. Thank you.
Yeah. Rich Condit, what do you have?
I ran across a thing that is Barack Obama, an article in Wired called "Now is the Greatest Time to Be Alive." And what it is an essay that introduces the November issue of Wired where Barack Obama is the guest editor. And
there's in scientific journals wasn't enough.
Yeah, and there's a nice video that goes along with this when he, talks about this. And basically, the really cool theme is that what he's saying is that it is the greatest time to be alive because of science and technology, that there's so much that we can do. And it's a very nice essay, and I haven't looked at the issue. I mean, how does this guy do all this stuff? It's amazing. At any rate, it was really nice.
Yeah. Remember, he hired Joe Handelsman to be, part of his science advisory team a couple of years ago, microbiologist, and he appreciates it. She said to me many times he really gets science and especially microbiology, and it's really good. We're going to miss that.
Yep.
Kathy, what do you have for us today?
I picked something from Acapella Science. I've picked Acapella Science in the past, but this one was particularly cool because he's singing all about CRISPR-Cas9, and it's really well done. And when you get to the end of it, you see that he actually has a section that you can go to the next video and watch how he put it all together. It's really quite amazing. And I had picked it in part because I knew that Dixon had just looked at a whole bunch of YouTubes about CRISPR-Cas9 because he told us that on TWiP. And he won't know that I picked it for my pick here, but he will know that I picked it because I emailed it to him directly. And so I'm hoping he'll take a look at this CRISPR-Cas9 YouTube as well.
You need to include a warning with this. After you've watched this, you will not be able to read the term CRISPR-Cas9 without this tune sticking in your head. It's really,
gone
really
from
catchy.
mine. It's gone from mine because it's been a couple of days now, so it will go away.
Oh, good.
It's
good.
to the tune of Mr. Sandman for all you old folks out there,
right?
Which is always one of my favorite tunes from way back. Very nice.
Do you do that in barbershop?
No, but that's a good idea. That would be a great tune for barbershop.
Yeah.
Yeah, on TWiP we did an episode where they used CRISPR very much in the way that we talked about today. They did a genome-wide CRISPR screen in toxoplasma, which is one of the parasites you can grow in culture. You have to grow it in cells, but you can grow it and you can do these sorts of things. And so he was watching videos to make sure he understood how CRISPR worked. And then when he came in the morning, he came in and he said, "Can you explain how CRISPR works?" Okay, Dixon. So it's going to be used for so many things. It's just amazing. That's pretty cool. What did I see here? Yeah, and there's some of the suggested videos. They of course, have Jennifer Doudna here because it's CRISPR. Yeah, that's pretty cool. My pick is an After Hours. Now, everyone on TWiV will remember episode 300. We all went to Washington, D.C., and as part of the Microbes After Hours series, we did that episode there. So that is an event that happens at night in the ASM headquarters at Washington, so people who live there can go see it. If not, it's live streamed. And I wanted to let you know about one coming up this week, Wednesday, October 19th from 6:30 to 8:30 Eastern. It's called The Necrobiome, Microbial Life After Death. Jennifer,
appropriate October episode.
Absolutely. The host is Jennifer DeBrune of the University of Tennessee. She's a professor there, and she's going to talk about what happens to us after we die.
Is she connected with that body farm that the author who writes those one-word books, the most recent one was about the military, but...
right. There was Body was one of them, I think.
Grunt and...
I don't think so.
No, she's not connected.
I don't see that in any of the material on her website. at the University of Tennessee in the Institute of Agriculture. But I think it could be interesting, so we'll put a link to that. It'll be live streamed. Of course, they also record it and release it later. And then we have a listener pick from Jolene, which was earlier. Here's my listener pick. I was recently reading through a hard copy of Microbe and came across a book review written by Dr. Dennis Mangan, director of Chalk Talk Science Project. His idea is to help scientists become better communicators, especially in the area of convincing the public that science is cool and important. In our local IUB chapter of SACNAS, Society for the Advancement of Chicano, Latino, and Native Americans in Science, I started a Chalk Talk series for graduate students. I did this shortly after I discovered that to get a faculty job, you have to do one, but ours is formatted a bit differently since it's to a general audience and with less intense grilling. The SACNAS membership draws from many STEM fields, so graduate students must explain their science using a chalkboard only to the mixed grad-undergrad audience in 10 minutes with 5 minutes for questions. We also use an anonymous feedback form as well. Dr. Mangan's ideas align with what the ultimate goal of our series is, to convince those outside your field that your science is interesting and worth pursuing. We might try to invite him here sometime, but it would be neat if you could do an interview with him. In the meantime, my pick is his blog. It's cool. Chalk Talk Science by Dennis Mangan. Communicating science to the public, a professional necessity, which I always tell people at the end of my talks, and I see them staring at me blankly.
So just to clear it up for everybody who might also know of this book and not be able to think of her name, Mary Roach is the author of the book Stiff, and also Grunt and Gulp. Okay. But Stiff is the one about what happens to you after you die. And I was pretty sure that she visited a body farm in Tennessee where they do research on what happens in the bodies after they die.
We'd like to give away the 4th copy, that's the last copy I have, of Marilyn Rusink's book, Virus, an illustrated guide to 101 incredible microbes. It's published by Princeton University Press. All of you have heard our contests before. Here's the last one. If you'd like to win a copy, we'd like you to write an original limerick or haiku about viruses. We're going to look at them and pick the best one, of course. And remember, we have a family audience here, so make it clean. Make it...
we don't want any of those bawdy haiku that you hear.
No bawdy. Right? Best original limerick or haiku about viruses. And again, if you've sent in entries before in this contest, you can enter again. If you want to send in 20 limericks or haikus, you should just send the best one that you think, but you could send in a couple. But we're not going to limit you to one. It could be anywhere in the world. And we'll give you a couple of weeks to do this because we know it takes time and you don't often listen. But send that into TWiV@microbe.tv and you get that.
And not everybody's Bob Dylan, so that's another reason why it might take time.
That's right.
Is there a reason why you mentioned him?
Because of the Nobel Prize, of course.
Are you surprised? I was.
I heard it when it was announced first thing in the morning, and I just went, wow.
It's pretty cool.
The times they are a-changing.
Yeah.
You know, the first thing I thought of, it's Bob Zimmerman.
Yeah.
Okay, that's pretty cool. That's TWiV 411. Anyone want to search for something? You can find it at iTunes and at microbe.tv/TWiV. And as always, we love getting your questions and comments. And you know what? You don't even have to have a question. If you just want to tell us who you are and why you listen, we'd love to hear it. I'm telling you, it makes my heart warm to hear what you folks are up to. Send it to TWiV@microbe.tv. Also, consider supporting our science shows. You can go to one page, microbe.tv/contribute, and there is information for Patreon, PayPal, credit card, and a link to our Cafe Press store, which also supports us as well. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
Although soon you won't be in Florida anymore.
That's right. I think one more TWiV from Gainesville, and then there's a couple of other TWiVs, substitute TWiVs coming up, and then the next will be from Austin.
So we'll say you're an emeritus professor, University of Florida, now from Austin, Texas.
Broadcasting from Austin, Texas, or something like that, whatever.
Yeah, well, we'll have to come see you in Austin sometime. And
all right, we'll have a party.
We'll have a party there. We'll have local TWiV. I'll bet there are more than one person there that would love to come to that.
Yep. And Andrew Wakefield's there.
Oh, good.
Sorry.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsors of this episode, CuriosityStream and Drobo. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.